Hello, everyone. Thank you for joining us today in this meeting to talk about synthetic biology platforms. So, welcome, everyone. Thank you for joining. And we're very excited to have you all here and also the speakers. My name is Patty. I'm part of the Design League staff, and I will be giving you a brief introduction today. And then I will leave Jake and Jeremy, that we're all very excited to hear, to talk a bit about the platforms they're going to talk about today. So, okay, so I will start with thanking our partners. So here we have Ginkgo, that Jake is going to talk a bit more about what they do. We also have Synlogic, that is a biotech company that is dedicated to transforming disease to treatment, biotherapeutics and stuff like that. We've had an online meeting with them before, so go and check it out in our YouTube channel after we finish this one. We also have in our partners Instituto Senai. Instituto Senai is a leader in research and development in Brazil, so thank you for your support. And we have also Instituto Tecnológico Bali. So I will show you a bit later a bit about their work. So yeah, thank you to our partners also for sponsoring this event. And I'm going to talk about iGEM Foundation. So this is who is organizing the event. So as you know, iGEM Foundation is an independent organization that it's nonprofit, and we're dedicated to the advancement of synthetic biology education. And we have a competition, a global competition with teams and a lot of countries participating around the world. And as part of iGEM Foundation, we also have this idea of local people working on solving local problems using synthetic biology as a framework. So what we've been working in the last few years is to help people in different communities to use the power of this technology to develop synthetic biology everywhere and by everyone. And as part of this mission, we are iGEM Design League. So this is our competition that right now is running through Latin America. We have 26 teams in the competition participating right now from 9 countries. So a lot of people working in different places. And our goals are to design to help Latin American students to kickstart their careers in synthetic biology. So we also have iGEM Academy. That's an educational tool that we use to inspire and help students learn about synthetic biology. So they work on this through the season. And also to prepare this next generation of talents to build the future in the bioeconomy in our region. So that's why we're here today talking about biotech platforms. So as you know, synthetic biology is the combination of the knowledge of these life sciences with the engineering part to help redesign and use biological systems to create services or products. So the difference with other technologies is how the implementation of this engineering framework or this engineering cycle help us to redesign for biology. And we're going to show you an example that is from Instituto Bali about what they're doing and how they're using this technology to do a project in the Amazon. And so that's very exciting.
Quando olhamos para uma floresta tropical como a Amazônia, é difícil colocar em números a grande quantidade de produtos e serviços que uma região como essa nos oferece. É como se estivéssemos dentro de um grande armazém. Aqui se pode ter serviços que vão desde a regulação climática até mesmo a produção de medicamentos. Hoje vamos falar de uma planta muito usada na produção de medicamentos e importante fonte de renda na região, o jaborandi. O jaborandi é um arbusto que cresce na parte baixa da floresta e é a única fonte natural de pilocarpina. A pilocarpina é o ingrediente tirado das suas folhas e que é usado na composição de vários medicamentos, como os que servem para o tratamento de glaucoma, onde é o principal medicamento, e também nos tratamentos de alguns tipos de câncer. O jaborandi verdadeiro ocorre somente em 3 estados brasileiros: o Pará, o Maranhão e o Piauí, e é uma importante fonte de renda para as comunidades locais. No entanto, o extrativismo desordenado e o avanço do desmatamento na região reduziram a ocorrência do jaborandi. Hoje ele é uma espécie ameaçada de extinção. Aqui no Instituto Tecnológico Vale, em Belém, juntamente com outros parceiros, temos desenvolvido pesquisas que contribuem para a conservação e o manejo sustentável dessa espécie. Nossos estudos começaram com o mapeamento das populações de jaborandi na Floresta Nacional de Carajás, um dos redutos atuais da espécie por ser uma área protegida. Nós também buscamos identificar plantas matrizes que produzem a maior quantidade de pilocarpina, o ingrediente dos medicamentos. Até o momento, temos identificado plantas com até o dobro dessa substância. É por meio dessas plantas matrizes que estamos direcionando a produção de mudas, tanto para reflorestamento quanto para plantio em sistemas agroflorestais na região. Para compreender como uma planta produz mais pilocarpina do que a outra, estamos buscando respostas no DNA da planta. O genoma completo do jaborandi está sendo montado pelo ITV em uma parceria com a Universidade de Copenhague, na Dinamarca. Juntamente com estes resultados, usamos dados de expressão gênica para identificar a rota de síntese de pilocarpina, ou seja, os mecanismos que a planta ativa para produzir este composto. A importância de descobrir essa rota é possibilitar o aumento da capacidade de síntese de pilocarpina, pois o jaborandi, quando cultivado, produz pouco dessa substância. Isso faz com que a gente precise continuar coletando jaborandi na floresta.
Yeah, so that's great, and that's a small idea and only one project that it's working in the Amazon, but there are many projects around the world working in synthetic biology. And I've seen that you are joining us from Mexico, Chile, Brazil, Spain. So welcome, and we're very happy to have you here. So without further ado, we just move on with Jake and Jeremy that we're excited to hear. So we will start with Jake today, and then we will hear from Jeremy. And at the end, we'll have a small time to ask questions. So if you have any questions, the public that and everyone that's in the live streaming, please leave your questions in the comments so we can pass it to Jake and Jeremy at the end. Yeah, and I think we can start now. So let's hear from Jake. Please, Jake, introduce yourself and go ahead.
Hi, everybody. Hi, everybody. What's up? What's up, iGEM Design League? It's great to be here. How's everybody doing? I'm watching the chat. I'm having a great time. It's fun to be here on YouTube. I love it. I love iGEM. Very excited. So my name is Jake Wintermute. I work at Ginkgo Bioworks. My job is called Developer Evangelist. And so that includes a lot of what I'm going to do today. So I'm a technical storyteller. My job is to talk to people about what Ginkgo's capabilities are, how to make use of those capabilities to build your own synthetic biology companies and your own synthetic biology products, and to have conversations like this one about what the future of synthetic biology might look like that is enabled by platforms like Ginkgo's and by technologies like Ginkgo's Foundry. So that is what I am excited to talk to you about today, and that is what I'm happy to get questions about in the chat. So with that, I'm going to share with you some slides. Let's see how that's looking. Yes, there it is. It's looking good. So I want to start. I just want to introduce Ginkgo Bioworks a little bit, because I know that not everybody is familiar with our story. but our story is your story. Our story is the iGEM story. So here I am showing these are four of our founders. This is Jason, Reshma, Barry, and Austin. In 2006, at one of the very first iGEM competitions at MIT. So they got their start as an iGEM team. Their iGEM project was called Eau de Coli, which was to genetically engineer E. coli to produce fragrance molecules. They were also one of the first iGEM projects to have the name of their project be a silly pun that includes the name E. coli. So we're very proud of that here at Ginkgo. So the Ginkgo story is part of the synthetic biology story, and it's part of the iGEM story. And so we are big iGEM fans. We love supporting iGEM teams, and I think we share a lot of iGEM's values and a lot of the sort of vision that iGEM promotes and teaches about building with biology and engineering biology. So, so that was 2006. Fast forward to 2021, and Ginkgo launched as a public company. So this is our IPO on Wall Street. We put a picture of a T-Rex on Wall Street. Grow Everything is our logo. We've grown up a lot as a company, and there on the left is that's Drew Endy, one of the early synthetic biologists, wearing his shirt from the MIT 2006 iGEM team. So it goes to show, a lot can change in 15 years. So we've grown up. We're now one of the flagship synthetic biology companies, the flagship platform companies for synthetic biology. We are, so the idea of Ginkgo. So what does Ginkgo do? So Ginkgo is an R&D partner for synthetic biology. Our technology is built around the Foundry, which is a large-scale platform for automation, DNA design, DNA synthesis, assays and strain testing, and process optimization and scale-up. So our customers will come to us with a need for a particular organism. We will genetically engineer an organism for our customers and license it to them, which then they will use to make their own products. So we work with customers all across any industry that biology touches. So we work with therapeutics makers, we work in antibiotics discovery, we work in enzyme discovery, we work in industrial biotech, we work in food. Anyone out there who wants to build with biology, who wants to use an organism to make a product or to design a therapeutic using programmed DNA, Ginkgo is there to provide that opportunity as a service to them. And so we are an ecosystem company in the sense that we succeed when synthetic biology as a whole succeeds. We want to be there to provide synthetic biology services to a wide range of companies across the entire synthetic biology ecosystem. And so when we come to iGEM, when we support iGEM, we do it out of love, but we also do it out of a business interest, because we know that iGEM companies that iGEM is going to be the source of synthetic biology leaders in the next generation. So it is going to be you, the iGEM leaders, who are very likely to be building out the next generation of synthetic biology companies, and Ginkgo wants to be there as a platform to support you in making those products. Our mission is to make biology easier to engineer. because the world needs more biology. So this is the moment in biology. This is the time to be a synthetic biologist and to build with synthetic biology. The need for developers in synthetic biology is very old, but what has changed today is that the tech has finally caught up to the need, and the investment has finally caught up to the tech. We can actually do it now. We can actually use biology to solve a lot of these very important global challenges. We can edit genomes, we can design proteins, we can synthesize DNA by the gigabase. Anything biology makes, we can grow it up and we can ferment it 10,000 liters at a time. So Ginkgo wants to be the place that is bringing together all of those technologies and making them available to companies that want to build with biology. What it means for you and for your generation is, I think, that we are at the beginning of a very pivotal moment in what it looks like to build a synthetic biology company. For a very, very long time, biological research looked fundamentally the same. So this picture that I'm showing is something like 100 years old. This is what Louis Pasteur's lab looked like. And the basic model for how to build a biology company has looked the same for a really long time. So you can call it the 10 postdocs for 10 years model. What do you do when you want to build a biotech company? You get some really expensive lab space, you fill it up with some really expensive equipment, you hire a big team of experts, and then you give them what is in many ways a very boring job of iterating over and over again on a particular genetic design in order to make it work. So it's a very laborious process, and it's a very repetitive process, right? There are parts of the synthetic biology design cycle that are very fun, that are very rewarding, intellectually stimulating. There are parts that are very boring and repetitive, and I'm sure that this is something that you are familiar with now as an iGEM team. For scientists, this means dedicating a lot of brain power to a process that's very laborious and repetitive. For entrepreneurs, it means that you have to own a huge amount of technical risk. This is a very expensive process, and it's a very slow process. And for investors, it means you have to put a lot of money in for a really long time. So as long as synthetic biology looks like this, synthetic biology is going to be fundamentally a closed process, a very risk-sensitive process, and we're going to be very limited in what we can build with biology, just because we don't have tens of millions of dollars for every R&D project. Everybody who wants to solve a local problem with biotechnology can't afford to, according to this model. And this is nuts. And this is nuts because there are so many possibilities for what we can build with biology. If you just look out there in nature, there's so much that nature can do. There's a million ideas out there. There's a billion ideas out there. The video that we just looked at, I did not know about that molecule. I did not know about that project. It would not have occurred to me. And there are many, many more like it. It's effectively blue sky. There's no limit right now to the number of things that we can build with biology, as long as the technology is there to make it possible. Platform biotechnology is what gets us there. The next generation of biotech companies are not going to look like 10 postdocs working in a very expensive lab space for 10 years. The foundry is the place where a lot of that repetitive, scaled up work is going to happen. Ginkgo can be the place with the heavy machinery and the specialists to do millions of biological operations. So in this model, we're thinking about 3 postdocs for 3 years. The developers are focusing on the most creative, the most exploratory early stages of the project. Then the project transitions to the foundry, where all the iterative improvements to the design are happening. So this is your design, build, test, learn platform. So if you're making a protein, it's on the platform. If you're going to optimize the yield of a metabolic product, that's on the platform. Any microbial phenotype, any human cell line, any function that can be encoded with DNA, the platform is the enabling technology that takes it from good to great. And the next generation of companies are going to be built with this capability in mind. I am very excited for iGEM Design League in particular, because I really think iGEM Design League is at the forefront of this new concept of biology. There is a very real opportunity for synthetic biologists in Latin America, for example, or in anywhere, like really literally anywhere outside of like Boston and a few other cities that have invested in that really expensive, really heavy infrastructure for lab space. There is an opportunity to leapfrog that technology and start building around the foundry from day one, and avoiding the need to build out that heavy infrastructure that is in many ways obsolete. And so I think iGEM Design League is the place where I want to come to think about what is the right relationship between a platform like ours and between developers like you. And so I'll give you this metaphor. Okay, how does it work? How does it work to develop for a foundry? How does it work for someone like you to build a company on Ginkgo's platform? So here's my metaphor. It's like a tree. I love trees. Trees are awesome. I try to use trees as a metaphor whenever I can. Foundry biotech is like a tree. The leaves and branches of the tree, that's you. That's your job. You're the developers. The ideation comes from you, and the products come from you. So when you say, okay, I need to make this particular molecule, and I'm going to use it as a therapeutic, it was your idea to make that molecule, and it will be you that takes that therapeutic to patients, right? It's your product. That's what the developers do. The foundry is the execution specialist that supports you. So we're the trunk of the tree. The foundry is there to iterate on that development process, to do those design, build, test, learn cycles at scale with a lot of automation, with a lot of data behind it, that is going to accelerate that R&D process for you to make that product. The trunk of the tree, the interface between the foundry and the developers, is, in my view, the most interesting space in biotech today. So the relevant questions are, how do you communicate your needs? How do you describe a particular organism that you need to make? How do you specify the performance criteria to say, okay, Ginkgo, I need to make a yeast strain that produces this protein with this enzymatic activity, and it has to be at this yield using this feedstock. There needs to be a kind of an interoperable language that allows developers to clearly understand what a platform is capable of delivering, and then to use those capabilities in surprising ways. So the sort of key unlock in platform biotechnology is when the capabilities of a particular platform are clearly communicated and documented enough that they can be used by developers in surprising ways. So that is what I think. So that conversation, I think iGEM Design League is the place where that conversation should start. and can happen. And I think that is what the future of synthetic biology and platform biotech looks like. And with that, I will stop sharing and I will say, what do you think? What do you think? What do you think? What do you think, iGEM? What do you think, iGEM Design League? And I look forward to taking your questions.
Yes, I think people it's trying to actually take in everything that you just said, Jake, because it's revolutionizing what's going on in Ginkgo. So I think uh, it's a new way of thinking and designing science, and this is something that definitely, hopefully, the students will have, and everyone that's in the public will have time to think about, to ask questions a bit later. So thank you very much, Jake. We'll move on with
Thanks.
Jeremy now, and then we will have the questions at the end. So I will, yeah, thank you. Welcome, Jeremy. So we'll talk a bit about the different platform now.
Hi, yes.
Different way
Thank
of
you, Patty.
designing.
Hello, everyone. Hello, iGEM Leagues. It is nice to meet y'all today. I'm also going to share some screens. Hopefully, y'all can see this. Yeah. Cool. Okay. So, yeah, very nice to be here with you guys today. I am really excited to chat with y'all. And this is actually one of my introductions to iGEM and the iGEM Design Leagues. So, super exciting for me. But I am Jeremy Matthews, and I am a bioinformatics engineer at FormBio. So we are also a platform styled company. But what my job is I help develop tools and pipelines, algorithms, really any sort of development, software development to aid and assist in biological problem solving, whether that be synthetic biology or really any track of biology. But just helping our customers figure out what biological problems they're trying to solve and how can I apply, computer science, engineering, and brand new biotech to this problem in order to give them a solution that is usable in their space. So that really is what Form is all about, right? We are about enabling insights and outcomes in your work as a scientist, as a biologist, or future work, right? For some of our students that are listening. But that's really what the core of Form is all about. But before I get into that, let me tell you about how I got here. So I am raised in Texas and I still live here. I did my undergraduate here at UT, and it was in biomedical engineering. And I was really excited about that at the time. It seemed really cool to think of the idea of combining technology and life science biology. And I just kind of dived headfirst into it until I graduated and realized I needed to get a little more specific for finding jobs and stuff. And that's kind of what led me to bioinformatics and computational biology. So I continued my learning and my educational background, focusing here in bioinformatics, and from there was able to jump over into academia, where I joined a bioinformatics core facility here at UT Southwestern, which is the medical center here in Dallas, Texas. And it was really exciting and a great way for me to just lay a large, solid foundation for building, skills and really digging into bioinformatics that I wouldn't really get anywhere else. from there, I got the opportunity. My boss decided to leave academia and was invited to join a brand new company, Colossal Biosciences, to start the bioinformatics side of their company. And she asked me to come follow her. And so I did. And it was exciting. It was a chance to work on brand new technology and cutting edge science. And so I did it. And from Colossal, we spun out FormBio, which is where I work now, which is really just the bioinformatics and the solutions, the software solutions to biology. So Form itself is a relatively new company. We were founded last year, but we, despite being really new, we do have a lot of experience behind all the areas that we bring to our company, whether it is bioinformatics, whether it's AI, ML, whether it's just our software engineering team. Every person, I think, at our company, and it's really cool to see, but every person is really a titan of their area and their field and really makes a huge difference, and that's kind of what these stats here are trying to symbolize for y'all, right? Is how much experience, despite us being a small company, how much experience we carry, and the background behind, that's essentially driving what we do here at Form. So being new, we have already made a couple of big waves in the industry and in the biology sector as a whole. We have partnered with Google, and we are now one of their premier partners, which means that we are one of the few companies that they have exclusively recommend for their health science and life science solutions. We are their multi-omics platform recommendation, and that is a huge really pat on the back to the FormBio team and what they're able to do. Then we also partner with PacBio, who some of y'all may know as a sequencing company and a biotech company out there, and we really have intended to become their bioinformatics solution as well, really giving them the expertise and insight to analyze the data they produce, quickly and efficiently, which I think all of their customers and consumers really love as well. And then finally, for just optimization standpoints, NVIDIA has been a great partner with us, where we have been able to integrate NVIDIA's technology and their expertise into our solutions that in turn help push science forward and really push forward the projects that get brought forward by people like you and others in the iGEM community. So outside of our big partners, we also have academic side partners, which are really partners for helping us test and push forward a lot of our solutions and things that we're able to do in the biological space. Some of these are partners that we've brought about either organically or through connections that we had from a lot of our scientists being from academia prior. But it's really a growing list, and we're hoping to expand to universities across the globe, because we really see Form being having the ability to give biologists the power of computers without needing to know computers full through and through. So where Form is really, is really able to help and make a difference is, applying our expertise and our assistance in the computational side of biology and allowing biologists and synthetic biologists like yourselves to focus on the science and really focus on solving the biological problems that you have. And we try and do that by touching a couple of different key aspects, but I think the three that are laid here are kind of areas that we are focusing on right now, especially early on in our company's life. And those are, first of all, just general computational research, right? We want our platform to be an easy entrance for biologists who have never played with computers or don't maybe have the computer infrastructure, right? We can easily, with a simple connection to a website, can empower biologists to do high level, high throughput experiments and analysis without needing all the infrastructure behind it, right? And I think that is a really cool aspect that has a big effect on iGEM leagues as well, and the design leagues, where instead of having to, like we said, put in all the infrastructure and money behind building out computer systems and labs and everything, we try to make that a little easier just with a touch of a button, using our platform to get that done. And then we also deep dive into cell and gene therapy and genome and genetics research, right? So this is where we go beyond the base level analysis, and we really bring our experience and our AI skills to try and solve large problems, right? And I will dive into each of these as we go, just to give y'all examples of what FormBio is doing and what we can do to help really shape the future of biology and synthetic biology. So with the areas that we want to touch, right, that what I'm showing here is just the range of expertise and the range of tools that FormBio can bring, right? So not only do we have workflows across a variety of different types, we also have multiple tools and back that with reports that are built for a biologist and made for a common biologist to be able to read and understand, right? We want to cut out the complicated computer aspect of things and make biology a lot simpler, right? So with that, right, I said we dive into each of these, right? So first off, I think a great place to start with synthetic biology is kind of where we birthed from, right? Coming from Colossal, which is a de-extinction and a species restoration, biodiversity sort of company, genome editing was really our first entrance into what Form was, right? And this was built around a lot of genome editing and validation for editing experiments, DNA synthesis and DNA design, right? And just being really the crux of synthetic biology. And so that being kind of where we started, right? Our solutions that we brought forward were, first of all, variant detection, a full encasing variant detection workflow that a biologist with a couple of points and clicks on a website can analyze samples against each other. They can determine mutations in their samples. They can determine genetic changes and rank these, rate these, and really understand what's happening in their sequencing, and that was one big step to gene editing, right? As you can imagine, discovering what to edit first is always a big key. And then along with building an amazing workflow, we then partnered with NVIDIA and sped sped the heck out of the thing. So it is really extremely accelerated. It is efficient, and it allows for high level analysis in fractions of the time that open source tools would take. The next step in our kind of gene editing idea was, how do we design edits, right? Guide design. There are tons of tools out there for guide design, but sometimes it needs to be a little simpler, right? Sometimes you don't know what editing strategy to use. Should you use HDR, base editing, right? Primate. There's so many different methods to genome editing nowadays that sometimes it's hard to tell what to use, and you just know, I need to make this edit to make the product that I need. So that's where we kind of started with developing our guide design tool, right? To be independent of editing strategies, independent of any sort of input, where we are able to give our users just the best method and best option for editing, regardless of what they're trying to edit, right? And then finally, we needed to validate this, right? So validating, it means after we make these edits in cells, how do we know that the edits worked, right? How do we make sure unwanted edits didn't happen? So we needed to really build out validation tools as well. So we now are able to validate, through multiple forms of sequencing. Sanger obviously is kind of the common, but NGS has really taken the stage with Amplicon-seq, which is targeted NGS, and then whole genome sequencing, which allows you to search an entire cell genome or organism genome to see targeted and off-target effects from editing, right? So these tools were kind of birthed out of that initial, how do we help Colossal solve their problems? And it was really cool to see the effect that form could make on these biological problems that Colossal was having, right? We were able to do variant detection extremely fast. Partnering with NVIDIA, we were able to bump it up to 88% faster. We were seeing an extreme drop in costs, obviously, because the less time you're using these computational tools, the less cost it is. So that kind of went hand in hand there. And then we also saw just an increase in our editing experiments that Colossal was running. We saw that our algorithm was able to increase their expected edits and increase the number of cells that were getting edited correctly, which was amazing to see, right? And it was a great pat on the back, if you will, for pushing us forward and showing us that there was really a space for us to build, right? And so off of that, we kind of dived into the market as a whole, right? We said we did great with Colossal, but we could do this with tons of other companies on a much larger scale. And so we turned to cell and gene therapy, right? And we really dove into this. And the example I'll go through here is our AAV optimization. So AAV is a viral, is a virus that can attack humans, but we have used it in therapeutics to make a viral vector, which essentially allows us to stick a piece of DNA inside this virus to have the virus put it into human cells. This is a common thing in cell and gene therapy. And we looked into AAV and said, how can we help improve this? How can we help the process, help with drug development? How can we make this cheaper for our end users to get cheaper drugs faster, right? And so with that problem, we developed FormSight. This is our highest level AI driven solution set for right now based around cell and gene therapy manufacturing. So FormSite is split into essentially two areas that we thought we could focus on. We've got Simulate and we've got Optimize. Simulate is, we essentially developed a large language AI model that is able to predict a construct and predict the output of developing a construct before actually testing it. So rather than having to spend time in a lab running each test, putting millions of dollars into each iteration, we are using AI to simulate that and to essentially cut out the need for any of that. So we can simulate and fast track to the best possible output to be manufactured. And then from there, another issue that was faced was, how do we then optimize that, right? How do we take the output and go another step forward? So we then turned our language model to optimizing this construct that was developed, right? So now we've not only cut out all the trial and error to build a construct, we're now able to use AI and optimize this construct to increase the success of manufacturing and to drop the cost, right? We're no longer trial and erroring our way through this process, which was costing, millions and millions of dollars only to drive the price of these drugs up worldwide for end users like us. So, with Simulate and with Optimize, we were really able to reduce costs, right? Save time in the manufacturing process, and along the way, we enhance safety, right? We were able to really separate out working constructs from constructs that didn't work, and we were showing that our predictions were significantly reducing the amount of time it takes to get these results, and for these drug manufacturing, cell and gene therapy manufacturing companies to move to the next step, right? And with Optimize, it really took that to the next level, right? As I stated before, this allows you to optimize the dosage, control your yield output for developing these drugs, and really reduce costs and save time, which is one of the biggest drives behind what we were doing, right? It saves infrastructure as well, because they no longer need to have all this pre-set up. So this AAV construct, when we applied it, we saw amazing results here as well. Reduced truncation of these constructs were decreased 70%, which originally they didn't even know why some of these truncations were happening, and there was no explanation. We were able to, using AI, give them the answers they needed and solve the problem of the occurrence of these truncations as well. We also increased just the amount of full reads by about 20%, which is an amazing increase for anyone that has ever played in the construct development. It was dramatically improved while, as we said before, reducing time and cost, right? Which in turn makes gene therapy solutions for you and me cheaper. Rather than being hundreds of thousands of dollars, maybe we can get it down to an affordable price. So that was kind of the drive behind our cell and gene therapy, right? So the final aspect that I'll dive into with Form and how we're trying to make waves in synthetic biology is the more base level, our foundation, right? We've got these amazing solutions for cutting edge CRISPR editing and DNA design. We've got amazing cell and gene therapy, AI driven solutions, but it's all built upon a solid foundation of just good old fashioned multiomics, NGS, biotech research and analysis. So this is kind of around three aspects of our platform that we give all our users access to. The first one being our workflows. These are multi-step large analysis pipelines that are all maintained up to date. We try and give as much and the absolute best type of analysis we can possible, validate them. These workflows include variant analysis, as I've discussed before. This includes chromatin structure analysis, that being ATAC-seq, ChIP-seq, cut and run, single cell analysis from single cell RNA, single cell attack, and we're constantly growing this list. The genome editing tools we have discussed with the CRISPR solutions. And then we do a lot of protein structure and protein design implementation of tools as well, to just try and give our user every possible workflow and analysis they may need to answer the questions, right, that they have about their biological data. Then we also provide a set of toolbox tools, which are not as computationally heavy as workflows, right? These are more quick one-off, everyday tasks. Align a couple of sequences, design some primers, transform annotation files, right? Translate or transcribe sequence data. These are just tools that help make your life easier, so you don't have to have 50 different web pages up with different tools. And finally, our Form Bench, which is kind of our gateway into the bioinformatics world, right? With bioinformatics comes the need to maintain your computer system, your computer environment, need to maintain packages and a bunch of CS jargon that a lot of times biologists don't want to deal with. But FormBench is like a beginner's guide to that, right? We host tools like RStudio, Jupyter Lab, which is Python, TensorBoard, right? CellXGene, which is a great single cell solution from the Chan Zuckerberg initiative. And these are, these tools are there for someone that maybe has some custom code they want to try and write, some custom graphical interface they want to build that isn't standard to the public, but they don't want to, host the entire thing on their own. So we really try and give the computational background needed to everyone that joins, and make sure that they have everything they need.
Hey, Jeremy, sorry.
Yeah.
It's been great. We are a bit over time, so I don't know if we can wrap it up to follow with some questions.
Let's we can skip this. This is just us partnering with another company to solve their issues. And then, what Form does, which is we just help optimize everything to give y'all better results, faster results, more publications, and more, external ability to play with your science and really empower yourselves. And that's all.
Great, thank you. So I will invite Jake back, and it's very interesting. I think when we talk about this is the century of biology and the biology revolution, we will never think about having biology without a lab and then from a computer. So this is a very exciting time for biology. We have a few questions in the chat, and I also have a few more questions that I received directly. So we will start with this question from Alejandra. That's for Jake. She's asking, so what is your vision for the future of SynBio and the next generation of talents? What skills do you think they need to learn to be ready for this bio revolution?
That's a great question, and I could give like a super long answer, of course, but since we're a little bit over time, I'll try to be as brief as possible. I think if there's one skill that I would recommend building in addition to your synthetic biology knowledge is product-based thinking. So I think the most valuable skill that biologists don't have today is the ability to identify products that the world needs and bring them to market in ways that people want and that people will pay for. So thinking about the product side of your technology, how are you going to produce it? How are you going to deliver it? What problem does it solve for people? Are there alternative solutions for that problem? What price does it need to reach in order to be competitive? Is it something that is going to be good for the world? All of that kind of thinking is not traditionally taught as part of a scientific curriculum, but it's absolutely essential if you're going to use synthetic biology to solve problems in the real world. And so I think that's the number one thing I would recommend for synthetic biologists who are starting out.
Great, thank you. Yeah, I think that's very important to go beyond what we learn in academics. So we have one question for Jeremy. Jonathan says, wow, what a great end-to-end tool from FormBio. Is this tool available for external research use?
So currently, I don't think there's a public link to the tool, but there are ways to sign up for it through the website by filling out your email and getting in contact with our team to get you onto the platform. Yeah.
Great, thank you. And I have a final question for both of you. Is these projects that our students and other people is designing from these different countries outside of the main hubs in biotechnology, how do they need to prepare to actually go into this new revolution, to work with these computational tools and go to look for biofoundries when we are kind of far away from, where this revolution is happening? So maybe if you want to start, Jake, and then we'll finish with Jeremy.
Yeah, I mean, so I think I guess maybe the short answer is I don't really know. It's an emerging technology, and it's not the kind of thing yet where there's a sort of a well-defined skill set that you need in order to be able to access foundry biotechnology. But some things that are clearly going to help: computational thinking, being really good at computational tools, understanding how to code, how to use AI, how to work with AI-based platforms. It's pretty clear that that's going to be central to the development cycle. Understanding automation, what automation is capable of, how to design a good automation experiment, and finding that correspondence between the data set that you need to solve a problem and the technologies that exist in biology to be able to create that data set. So high throughput thinking, that's clearly going to be a big part of it. And then I would also add, just to product-based thinking again. I think that I think biology is going to get easier. I think that it's going to get easier, and so as it gets easier, the importance of having unique access to a really cutting edge technology that's going to become less important, and being able to identify a really good problem is going to be more important, because this problem space is going to open up. So computational thinking, high throughput thinking, thinking about product thinking, and whatever the last thing was that I said. Finding problems, identifying problems.
I think that is probably an excellent answer to the question. I think outside of what Jake has already stated, I think other great opportunities are kind of what y'all are already doing, right? Partnering and finding things like iGEM, right? And things that can help bring you to the technology and the technology to you, right? That is always going to help push things forward, right? But yes, understanding, right, a little bit of code, understanding, right, because it's not, I think the world is no longer a one track sort of destination output, right? It is, even in biology now, it's know the biology, know some coding, know some analytics, right? I think to add on to Jake, I think me personally, statistics and math are a huge part as well, right? People don't always think about biology in terms of statistics, but it was my training, and that's kind of how I look at biology, and it gives you a different insight, right? So I think it's that not being afraid to try and go bug someone, right? I think that was one of the best advices I got for just building myself in this field is don't be afraid to go ask, right? Email, cold call. Worst that happens is they say no, right? And you're just back to where you were. That nothing negative there. So yeah, I think those would be my advices to the people that really want to try and bridge that gap there that exists. But as Jake stated, give it another 5-10 years and the gap will be smaller and we'll be looking at a new gap, right? So there's always something around the corner. So
Yeah, that we need to keep working on. Great, thank you both. So yeah, we're a bit over past the time, so I'm going to close now. I want to thank you very much to both Jake and Jeremy. FormBio and Ginkgo have been great supporters, and thank you for bringing this vision and help us open up a bit the panorama for our students and people in Latin America. Everyone who is there, please remember that this is going to stay recorded in our YouTube channel, so you can watch it again, and you can watch it again as many times as you like. And you can also go back and watch the meeting we have for health in synthetic biology and also for sustainable technologies. So thank you, everyone. And that's it for today. See you then. Bye-bye.
Thank you.
Thanks, everybody.
Bye.